Continuing on in our summary of the Fraxa investigators meeting, day 2, mid-morning, Bob Wong, SUNY Downstate in Brooklyn. He's one of the world's leading authorities on the role of mGluRs in epileptogenesis. And he's actually been studying an interesting phenomenon that applies not just to Fragile X, but in general, seizure disorders and a lot of other potential applications. But in studying mGluR5, he found that when you apply agonists, like glutamate, to the receptor, it enters a kind of persistent upstate. And then when you apply antagonists, like MPEP or the mGluR5 antagonist that we're talking about, not only are you blocking the activity of the receptor and the response to neurotransmitter, but you actually switch the receptor into a kind of persistent low functioning state. So this is very different from our usual view of how receptors work. We usually think that an antagonist binds to it and that just kind of blocks the function as long as it's around, and then it reverts to normal. But what he found was it doesn't revert to normal. It stays down even after the antagonist is gone. So this explains, I think, part of the therapeutic mechanism of action of mGluR5 antagonists, and it kind of answers the little bit of a riddle that had been posed early on, which is how these antagonists could have such rapid effects in the Fragile X mouse model when they're applied, because the conventional thinking is that you block the receptor and it blocks the protein translation. It takes a while to really have an effect, but what we were seeing was it has an almost instant effect that's persistent for a long period of time. And that's really going to be critical going forward as we think about the mechanism of action of these drugs in Fragile X. I thought it was a fascinating series of experiments that Bob did. I think a lot of the people who aren't really into mGluR5 kind of didn't necessarily get what he was saying, but it's really another one of these revolutionary findings that changes your way of thinking about things. So it's really cool. Eric Klan followed Bob, and he's talking about these same signaling pathways, and a little bit of I describe it as a minor controversy. A friendly difference of opinion seems to be evolving in the role of the specific signaling pathways. We tend to talk about signaling pathways coupled to mGluR5. It turns out there are many signaling pathways that are coupled to all of these receptors, and to make it even more confusing, a lot of the elements, the specific enzymes that are in these signaling pathways, are shared by multiple receptors. So a lot of the same things that are used to cause long-term depression cause long-term potentiation, and it's not exactly clear which things are really important and which aren't. But in Fragile X, one of the minor controversies is the role of this so-called mTOR pathway, and that stands for mammalian target of rapamycin, MTOR. And mTOR is of enormous interest to the autism researchers, because it's known that several genetic forms of autism involve dysfunction in this pathway, in particular tuberous sclerosis and a number of rarer genetic conditions, but also Fragile X. But Mark Baer has actually just recently announced, including at this meeting, that in fact the effects of Fragile X and tuberous sclerosis on the mTOR pathway actually seem to be completely opposite. So we do have a kind of final common pathway, but they're not final in the same direction. They're final in opposite directions. So we're starting to delineate what is actually different between the different forms of autism, especially these genetically caused forms of autism. And the bottom line is that if you have a dysregulation one way or the other, it could be bad for you. So if you have way too much or way too little function in any of these signaling pathways, in either case, the signal is lost. So we're starting to get a better sense of that, and Eric talked about some of the specifics and how different members of this pathway are phosphorylated, turned on and off. followed by Suzanne Zukin, who's one of the people that collaborates with him very closely, with further discussion of that, and as well as Flora Tassone from the Mind Institute, also talking about that, and she's actually seen some alterations in Fragile X, the Fragile X Prader-Willi phenotype, and actual Prader-Willi syndrome as well. So we're starting to figure out what the final common pathways are that connect different forms of autism. Tom Dockendorf talked a little bit about the effects of the different isoforms of Fragile X protein. And that's one of the things that lends another layer of complexity to the study of Fragile X, is that this missing protein, FMRP, can exist in several different forms. And it seems pretty clear that these different forms are produced under different circumstances in different places, and they have different functions. And so you can produce an alternate form of the protein to do different things when a cell is in a different state. Certainly different tissues produce different forms of the protein as well. Carolyn Beebe Smith then followed that with a really interesting discussion of human PET scan data. She's actually managed to scan a fairly large number of Fragile X subjects with for protein synthesis rates in a PET scanner, and you can actually visualize the rate of protein synthesis in the brain as it's happening. That is important because we have seen from the previous studies that dysregulation in the rate of protein synthesis is really critical to the pathology in Fragile X. And potentially this kind of study could be used as an outcome measure in a drug trial. The key is to see if you can actually do it in Fragile X people. And so far so good. Results are not in yet because the study is still ongoing, so they haven't started analyzing the data yet. But they do have some excellent results from the knockout mouse that show especially particular brain regions have increased rates of protein synthesis, and you can see it on a whole brain level. Presumably most of the excess is in the dendrites, but it turns out dendrites constitute in most neurons something like a third of the whole cell mass, so it's a significant part of the brain, and you can actually see the difference on a PET scanner. So it'll be nice to develop this technique a little bit further and see if it can be used as an outcome measure or as a way of understanding in particular what brain regions are most important in Fragile X. As we moved into the afternoon session, we heard quite a few discussions about different areas of the brain and how they were involved in Fragile X, people looking at inhibitory networks of proteins where there seems to be a general downregulation of inhibition. So the cells that are in charge of keeping all the other parts of the brain in check, so you don't have runaway electrical activity like a seizure, for example, or milder forms of runaway electrical activity like hyperactivity or difficulty sleeping. That inhibitory network is definitely a little sluggish in Fragile X. And it's not necessarily because there's anything wrong with those cells. It seems to be actually down-regulated. So all parts of that system are being told to go slow, inappropriately. So that means all the machinery is in place. It's all working. It's just the machine is telling itself not to activate that system. So that is a potential therapeutic target, and it's something that some of these researchers are looking at. We also have some folks that are looking at that system in the amygdala, the emotional parts of the brain, and they're finding some definite room for improvement there.